So moving on to our topic, in between, I must tell you about a learning platform, that is the Unacademy. You can boost up your preparations for the various competitive examinations with the help of the Unacademy. You can join the Unacademy Plus subscription as well as the Iconic subscription for various exams like the GATE Excel, GATE Biotechnology, and the GATE ESC as well as the CSIR NET exam. They also conduct the All India mock test exams for the GATE 2020 for every Sunday from January 2 to January 30. You can enroll for this particular mock test for various exams, and for the GATE aspirants, the link for the enrollment I will give in the description. At Unacademy, you can clear all your doubts at a live time. You can raise a hand in between a class, or you can talk to your educator in a live class by raising a hand. So it will create a classroom-like environment on the platform, on the online platform also. You can also ask your doubt even post class. आप class के बाद भी अपना doubt जो है वो clear कर सकते हैं. You can take a picture of the doubt, or you can type the doubt and can send to your particular educator. If you are an Unacademy Plus subscriber, you can ask the doubt anytime on the Unacademy. Unacademy provide interactive live classes. They also generate polls for the learners where you can clear your doubts as well as the group discussions. You can never miss a class. You can get a recording of the class later as well. And you're also getting alerts or the notification for the class for a particular time. You're also having the PDF notes for any concerned topic or any class, and anytime, anywhere. You can access all of the recordings. If you're a Plus subscriber, you can access all of the educators and their recordings anytime to prepare your notes and to get their PDF notes. So these are also some perks that the Unacademy provide on the learning platform. Along with the Plus subscription of the Unacademy, they also offer the Iconic subscription where you can access your educator, or you are having access with your educator in a live time where you can clarify your concepts, you can discuss your doubts, and you can have the live mentoring sessions for the exam strategy, even in the Iconic subscription. And in the Plus subscription, you can access all the perks that we have just discussed, like live classes, video recordings, as well as the PDF notes. The GATE ESC aspirants can join the new batches for the GATE 2K23 and 2K24. The batches are starting from the 19th of the January. So all of the aspirants which are targeting the 2023 and 2024 GATE, they can join the various branches according to their concerned branch, like mechanical, civil, or the electrical. Using a code YG10, you can join the Unacademy Plus subscription and you will get a 10% discount on your subscription. So let's just start with our.
Hello, welcome to the YouTube channel Botany for All. In today's video, we are going to discuss about an easy and important topic in the reproduction of flowering plants, that is the megasporogenesis. So this topic, megasporogenesis, is consist of the two words. One is the megaspore and another is the genesis. So megaspore is a kind of noun, or it is a word that we are going to discuss, that what is the megaspore, and genesis means the production. The literal meaning of genesis is production. So the production or the formation of megaspore, that process is called the megasporogenesis. During or in the reproduction of the flowering plants, the angiosperm or flowering plants is an important division of the plants and the most modern division. In the flowering plants, there is a particular structure which is called the flower, which is a special feature of the angiosperm plants only. Earlier plants like pteridophytes, gymnosperm, algae, or even the bryophytes, they are not having the flowers. They are having the sex organs, they are having the reproduction, but they are not having the flowers. It is a characteristic feature of the angiosperms. So in any angiosperm plant, there is a haplodiplobiontic life cycle, or we can say the alternation of generation. There are two phases. One are the gametophytic phase as well as the sporophytic phase in the complete life cycle of a plant. So there is an alternation of generation. So generally, a plant which we see, okay, let's say a mango tree. A mango tree that we see is a sporophyte and the ploidy is diploid. So the sporophytic generation is dominant in the angiosperm plants, whereas it is recessive in the algae as well as the bryophyte. In algae and bryophyte, gametophytic phase is dominant, and in the another divisions like pteridophyte, gymnosperm, and the angiosperms, the sporophytic generation is dominant. So the plant which we see is a sporophyte having a diploid ploidy. So this sporophyte will have sporophylls. So the sporophyll, micro and mega. The microsporophyll and megasporophyll. The mega and micro generally represents the size of the spores or the sporophylls, and usually the female spores are larger. That's why megasporophyll represent a female sex, and the microsporophyll or the microspores represent a male sex. So in a plant generation, microsporophyll or the microword represents the male generation, and mega represents the female generation or the female sex. So megasporophyll as well as the microsporophyll are arranged in a special structure which is called the flower. So sporophyte will have the flower. Flower will also be diploid. All of the things that are present in the flower, all of the tissues, all of the cells, they will be diploid. So in a flower, there are two kinds of the sporangium. If a flower is unisexual, the sporophylls or the sporangium will also be differentiated. If a flower is bisexual, having both sexes at the same flower, it will having megasporangium as well as the microsporangium on the same flower. That we will discuss in the upcoming videos. So if a flower is bisexual, it will having both of the sexes. So it is having megasporangium, the female one, and the microsporangium. So these sporangiums will be also diploid. Since they are sporangium, so they will contain the spores. The megasporangium will produce megaspore, and microsporangium will produce microspore. Be remember, these are spores. Okay, so they will not directly fuse. Spores are haploid, whereas the sporangiums were diploid. It means during the formation of spore, the main topic, the core topic that we are going to study is the megasporogenesis. So here is the link. The production of megaspore from the megasporangium, it undergoes meiotic division or the meiosis. So this process, the megasporogenesis or microsporogenesis that we'll discuss in the upcoming video, it is a meiotic division. In the megasporangium, there are megaspore mother cells. Similarly, the microspore mother cells, the megaspore mother cell. This cell is diploid, the mother cell, which will produce megaspore, and the megaspores are haploid. So after the megaspore mother cell, there will be meiosis. So the last diploid generation in a plant is the megaspore mother cell, after which the meiosis occurs and megaspores are produced. And this process is called megasporogenesis that we are going to discuss now. Now the spores will not fuse. The spores will produce the gametophytes. We are only talking about the megaspore, so it will produce the mega gametophyte. Because megaspore is haploid, gametophyte is also haploid. Gametophyte will also not fuse. Okay, mega gametophyte will produce female gamete or the mega gamete, that is the egg. Egg is also haploid. Egg will meet with a sperm or the male gamete. Similarly, microspore is going to produce the microgametophyte, in which the microgametes means male gametes will be produced, and the gametes, haploid and haploid. The gametes will fuse. This is called fertilization. Although there is a special kind of the fertilization in the angiosperm plants, which is called double fertilization, that we are also going to discuss in the upcoming videos. So after the fertilization, egg and male gamete fuses to form a diploid structure that is called the zygote. So again, the sporophytic generation or the diploid generation started. So this zygote develops into the embryo, which is also diploid. Embryo is developed inside the seed. Seed is also diploid, and a seed is germinated into a new plant. The plant is also diploid or the sporophyte. So this is a kind of summary of the life cycle of an angiosperm plant. Particularly, we are here that we are studying the formation of megaspores, a haploid megaspore from a diploid megaspore mother cell. So how this process is initiated, that we are going to discuss. So in the female reproductive part, suppose in a flower, the female reproductive part is called the gynoecium or the gynoecium. The gynoecium having a unit that is called pistil. Pistil having three things: a stigma, that is for the reception of pollen grains, a style which leads the pollen grains up to the ovary, and third one is the ovary. Kind of this one. This hollow structure is the ovary. This is style, and this is stigma. So in the ovary, there are megasporophyll containing structure which are called the carpels, and in the carpel there are the ovules. Suppose there are two carpels or the two chambers. Generally, the number of carpel is similar to the number of chambers or the locules, but not always. Okay, so generally it is happen that number of carpels is equal to the number of locules. So suppose this is one carpel, this is another carpel. So in each of the carpel there are ovules. Let's say these are the ovules. So ovary is diploid, ovules are diploid. Now let's just focus on a single ovule. How a single ovule is, how the structure of a single ovule is there. So ovule is having different layers and a mass of cells. There is a central column or a hollow column filled with a large number of cells. That here I am drawing those cells. Cells are not having intercellular space. They are intactly arranged. This is a single ovule. And there are many number of ovules. There may be many ovules in a carpel. And even the number of carpels may be many in an ovary. And ovary is a unit of pistil. And pistil is a unit of female reproductive part upon the flower. So we remember the hierarchy. We are in the flower, pistil, ovary, carpel, and the ovule. So this central mass of the cells is called nucellus. Each and every cell of the nucellus is diploid. Okay. Ovule having central mass of cells, which is called the nucellus. There are various layers which are surrounding the nucellus, which are called integuments. The attachment point is called a funicle. There are two ends of the ovule. This is the micropylar end. This one is the chalazial end. There may be vascular supply, okay, like xylem and phloem, which will supply the different nutrients or the hormones and the several other things like water also. So this is the kind of typical ovule. So the main concern is over there, this compact mass or the large number of cells which are present in the center, which are called the nucellus. Each and every cell of the ovule is diploid. The integuments itself are also diploid. Okay, several other cells are also diploid, and mainly the nucellus are also diploid. So we are discussing the megasporogenesis. So till now everything is diploid. In the same video we have discussed that megasporogenesis is the formation of megaspore from the megaspore mother cell. There would be a diploid megaspore mother cell which will form the megaspore or the functional megaspore, which will be haploid, and generally the process will be a meiotic process. So there will be occur the meiosis, and the process is called megasporogenesis. So in the ovule, in the nucellus, one cell, okay, towards the micropylar end, one nucellus cell will undergo active cytoplasmic division, active nuclear division. Okay, it will become more prominent. Prominent means it will become more active. So it will increase in size. There will be several changes in a particular nucellus cell towards the micropylar end or the hypodermal end. So it will increase in size. Metabolism will increase. Cytoplasm increase, become dense, and the contents increase. Okay, so that particular cell which take the responsibility to initiate something among all the nucellus cells towards the micropylar cell, that single cell will be called archisporial cell. Archisporial cell. This is the first step. We remember among all cells of the nucleus, one cell takes the responsibility to initiate the process of megasporogenesis. That cell will be called archisporial cell, and that cell will also be a diploid cell because it is formed from the nucellus itself. Now remaining cells are nucellus itself, and this particular cell which increased in size or the metabolism increases, that will be called the archisporial cell. Okay, so now see, this was the ovule. Here we have discussed that one cell. These are the remaining nuclear cells. This one cell was archisporial cell. Okay. Now this archisporial cell will divide transversely and mitotically. So it will divide to form the two cells. Here I am representing this thing. The archisporial cell itself divide mitotically, or just a simple cell division, and form the two cells. The inner one, this was the micropylar end, we remember, and this was the chalazal end. Okay, so it divides. So the cell, the new cell formed towards the micropylar end will called a parietal cell, and the inner one, inner means towards the chalazal end, that cell will be called a primary sporogenous cell. This is called primary parietal cell, and the towards the chalazal end, the cell will be called primary sporogenous cell. So this division is mitotic division, and it is formed from the archisporial cell itself. Now this parietal cell will form several layers, will form the several cells which will form a parietal layer like this. It divides longitudinally. This parietal cell also divided now by a simple cell division or mitotically, and form the several cells which form the parietal layer towards the micropylar end. Okay, this sporogenous cell divide meiotically. Now it will divide meiotically, and it will behave like a megaspore mother cell. Megaspore mother cell. Now see, which cell is the megaspore mother cell? First, a cell takes a responsibility to divide among all the cells of the nucleus. The nucleus was diploid, archisporial cell was also diploid. Archisporial cell divide mitotically, so it will form a parietal cell as well as a primary sporogenous cell. Primary sporogenous cell is also diploid, parietal cell is also diploid. The parietal cell, the diploid parietal cell, divide mitotically and form several diploid parietal layers. Okay, whereas primary sporogenous cell, which is also called megaspore mother cell. So this one is also diploid because till now there is no meiotic division. So this primary sporogenous cell is called megaspore mother cell, and now it will divide meiotically. From here it will divide meiotically. Okay, so this particular cell, the megaspore mother cell, divide meiotically and transversely. So 1, 2. In the meiosis 1, it formed the 2 cell. Only the megaspore mother cell, the primary sporogenous cell itself, forming a 2 cell by meiosis 1. And in the another meiosis, it forms total 4 cells. So from a single megaspore mother cell, total 4 cells are now produced. And this process is called megasporogenesis, and it is a meiotic process. So one mother cell, one megaspore mother cell, this is one, not primary, which is diploid, divides meiotically to form 4 cells. Now see, we won't say that these all 4 cells are megaspore. What happens is toward the chalazal end, okay, only one cell out of these 4, only one cell, which is the outermost or outermost towards the chalazal end, that remains only active or alive. Other three towards the micropylar end, they degenerate. Okay, so what happens is there is only one functional megaspore. There is only one functional megaspore remain. The other 3 degenerate. Although from one megaspore mother cell, by one meiosis, since there is one megaspore mother cell, one meiosis, 4 cells were produced. But all these 4 cells are not megaspore because only one, which is towards the chalazal end, remains active or remain functional. The other 3 towards the micropylar end degenerate. Okay, so final result is what? The final result is that from one megaspore mother cell, which is a diploid cell. Okay, the process is megasporogenesis, and it is a meiotic process. Okay, only one megaspore is produced. So sometimes questions are asked like that how many meiotic divisions are required to form 100 megaspore. To form 100 megaspore, how many meiotic divisions are required from 100 megaspore mother cell? So it will be 100 meiotic divisions, because from one megaspore mother cell, only one megaspore is formed. Whereas in the microspores or the microsporogenesis, from one microspore mother cell, 4 microspores are produced. From one microspore mother cell, 4 microspores are produced. Okay, so only 25 meiosis, because one microspore mother cell is producing 4 microspores. So 25 meiosis will produce 100 microspores. So for producing 100 microspore, only 25 meiosis are required, because the count is 4 times. But here in the case of megaspore, for the production of 100 megaspore, 100 meiotic divisions are required, because one megaspore mother cell is generating only one megaspore. So it was a quick process or a detailed process of the megasporogenesis. It was started with the archisporial cell, that in the ovule there are nucleus which are diploid. One cell from the nucleus become archisporial cell. The archisporial cell divide mitotically, form 2 cells. One the primary parietal cells toward the micropylar end, and one towards the inner end of the chalazial end, that is the primary sporogenous cell. This primary sporogenous cell is called the megaspore mother cell. Now it will divide meiotically. In the meiosis, it will produce 4 cells. This is called a linear tetrad, because all the 4 cells are arranged linearly. Linear tetrad. These 4 are not megaspores. The 3 towards the micropylar end degenerate. Only one remains functional, that is towards the chalazial end. That one functional cell is called megaspore. So it is haploid. Although these all 4 cells were haploid because they are produced by the meiosis, but they degenerate. 3 of the cells degenerate. So only one becomes the functional megaspore. So it is the functional megaspore, and the process is called the megasporogenesis. There is another case that sometimes this archisporial cell directly functions as megaspore mother cell. So there is no problem. Archisporial cell was also diploid. Megaspore mother cell is also diploid. So if the archisporial cell is directly acting as a megaspore mother cell, then this step will be eliminated. That it will not form the parietal or the sporogenous cell. It will directly form the sporogenous cells or the spores. So it may divide mitotically to form parietal as well as sporogenous cells, or it may directly act as a megaspore mother cell. So it is all about the megasporogenesis. In the upcoming video, we will discuss about the megagametogenesis, that how this functional megaspore will produce the gametophyte, the megagametophyte, or the female gametophyte, which is generally called the embryo sac. So that's all for today. Have a nice day.